Good afternoon, Good afternoon, ladies and gentlemen. My name is Sophia, and I will be discussing on how to isolate enzymes and check their efficiency. And as Ian described, we isolated cellulose degrading bacteria to get their cellulases. I will describe how we isolated enzymes and check their efficiency. So, what are enzymes? Well, enzymes are catalysts that reduce the activation energy of a chemical reaction. Most are proteins. We all have enzymes in our bodies. As we have been discussing, we are trying to isolate cellulases, enzymes that can degrade cellulose. Okay. So what we did in our project was we plated a bunch of cells in liquid media, LB. We did this because in liquid media it's a better environment for them to reproduce because of the nutrients that they encounter in the media. However, when we did this, the bacteria stopped producing the cellulases, the enzymes that can degrade cellulose, because they had nutrients. So basically, they got lazy. They didn't do the enzymes no more because they didn't need it because they had bacteria, plenty of bacteria. So then we plated the bacteria again in powdered cellulose in plates. And we did this so the bacteria can produce enzymes again and for other techniques. So this has a lot to do with the project, and I'll explain shortly. So some enzymes are extracellular. What I mean by extracellular is that in the bacteria's genome, they're outside the bacteria's genome. That's what I basically mean, that they're outside. So if an enzyme, for example, if we wanted to, we can't eat a cow. We can't eat a whole cow. Even if we wanted to, we can't. So we have to chop it, we have to degrade it, we have to kill it and whatnot, so it can finally get into pieces where we can eat it. So the same for the bacteria. The bacteria has these enzymes outside of the cell, so the enzymes can break down the cellulose, so then the bacteria can digest the cellulose. Because bacteria can't, just like we can't eat a whole cow, the bacteria can't eat cellulose, a whole piece of cellulose, because cellulose is a very rigid structure. So, and this is good in our experiment that enzymes are extracellular. Well, the ones that we're studying are extracellular because we wanted to filter the enzymes. That's one of the reasons why we plated bacteria in cellulose powder plates, because we wanted to filter them for other techniques that I'll mention shortly. So how do we harvest these enzymes? Well, it's like orange juice. It's like, for those who don't like orange juice, you basically pour the orange juice, you leave the pulp, and you grab the orange juice free with pulp. So that's what we basically wanted to do. We wanted to pour the powder with the bacteria, and then we poured a buffer and just grabbed the enzymes and left everything else there. Okay, so just like in school, we get tested, right, to see if we're learning. Well, an enzyme assay is a test we run on enzymes to see if they can degrade, in this case, cellulose, in an efficient manner. So basically, this is a cellulose, in the assay we use the cellulose structure with dye on it. And it had dye attached to it every 20 monomers. And we wanted to just see if the cellulase can cut the cellulose. And we measured this by color depth. We can see the enzyme is doing its job if the blue dye shows more. Notice how the cellulose is right here because it's blue and it's dyed with blue. cellulose is insoluble. But when the enzymes cut it, the monomers float up to the solution and it becomes soluble. And we notice this by color. The more darker it is, the better. In our enzyme assays, we noticed that without purifying the protein, there was still some activity. You can see this in the graph from our bacterial strains. We got the enzyme for bacterial strains. We then run a protein gel. A protein gel is a technique used to separate and characterize proteins. We wanted to evaluate the purity of our enzymes, and we can see that our protein was about 33 kilodaltons. We will next what we will do next is do chromatography to purify our protein because we want a pure enzyme. So once we have a pure protein, we're happy because we got the pure protein, we got the enzyme from the bacteria, and we identified the strain. So now we're ready to sequence and analyze protein even more. And Vernon, and my colleague Vernon will talk about that shortly.